Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman-Marquis, Dr. Scientist, and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptides series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, Humanin. And this is a compound with an origin story unlike any other on this list, because it wasn't found in a longevity screen or a performance lab or a sports medicine clinic. It was found in the brain of someone who had died of Alzheimer's disease, in the one part of that brain that had, against all odds, survived. In 2001, a group of Japanese researchers led by Yuichi Hashimoto and Ikuo Nishimoto were trying to answer a specific question. In Alzheimer's disease, neurons die, but not uniformly. Some regions are devastated, others are relatively spared. The occipital lobe, the visual cortex at the back of the brain, tends to hold up comparatively well, even in advanced disease. The team's reasoning was elegant. If some neurons are surviving in a brain where others are dying, perhaps those surviving cells are producing something protective. Perhaps there's a factor in there that's keeping them alive. So they built a cDNA library from the occipital lobe of a patient who had died with Alzheimer's disease, and they screened it, looking for genes that could protect cells against the neuronal death caused by Alzheimer's-related insults. And out of that screen came a short open reading frame encoding a small peptide that did exactly that. It rescued neurons from amyloid beta toxicity. It rescued them from the death signals associated with the disease. They named it Humanin, not after a Greek fate, not after a molecular feature, but as a statement of hope, a peptide from a human brain discovered in the search to defeat a disease that erases what makes us human. It's an unusually sentimental name for a molecule, and the researchers meant it. But the more remarkable finding came when they asked where this peptide was actually encoded, because the answer rewrote a piece of textbook biology. The Humanin sequence wasn't in the nuclear genome, where we expect our protein-coding genes to live. It was in the mitochondrial genome. Specifically, it was hidden inside the gene for the 16S ribosomal RNA, a region called MT-RNR2, a functional, secreted, bioactive peptide encoded within mitochondrial DNA. Humanin became the first known member of an entirely new class of molecules, the mitochondrial-derived peptides, and since 2001, that class has grown, and the idea it represents, that our mitochondria are not just power plants, but signaling hubs, sending peptide messages to the rest of the body, has become one of the more genuinely novel ideas in aging biology. So let's go through it properly. What Humanin actually is, what it does at the molecular level, what the human data shows, and what the honest picture looks like for anyone in this field. First, the basics, because the form and the origin of this peptide are unusual enough that they need spelling out carefully. Humanin is a small peptide, and I do mean small. Depending on where it's translated, it's either 24 amino acids or 21. That difference is not a rounding error. It reflects two different translation systems. If the mitochondrial ribosome reads the open reading frame using the mitochondrial genetic code, you get a 24 amino acid peptide. If the same messenger sequence is exported and read by the ribosomes in the cytoplasm using the standard genetic code, you get a 21 amino acid version. Both are biologically active. This dual origin has been a source of genuine debate in the field. Where is the physiologically relevant humanin actually made? And it's one of the reasons the peptide has been trickier to study than its size would suggest. The key structural point is that humanin is encoded in mitochondrial DNA within the MTRNR2 gene. Your mitochondria carry their own small circular genome, a relic of the bacterium that took up residence in our ancestral cells around 2 billion years ago. For decades, we thought that genome coded for just 13 proteins, all of them components of the respiratory chain. Humanin was the first evidence that the mitochondrial genome also encodes signaling peptides, molecules whose job is not to generate ATP, but to communicate. That reframing matters because it positions the mitochondrion as a sensor, something that monitors cellular stress, and when things go wrong, releases peptides like humanin to coordinate a protective response. And that's the essential character of this molecule. Humanin is a cytoprotective peptide. Its central, most reproducible function, the one it was discovered doing, is keeping cells alive under conditions that would otherwise kill them. It acts both inside the cell and outside it as a secreted, circulating factor. And like clotho, which we covered previously, it's a molecule whose levels in human blood decline substantially with age. The reason Humanin has held research interest for over 2 decades is that it interfaces with several distinct biological systems: apoptosis, metabolism, and inflammation, through mechanisms that are, in most cases, genuinely well characterized at the molecular level. Let me go through the major ones. The first and the most fundamental is the suppression of apoptosis, programmed cell death. Apoptosis is governed in large part by the BCL2 family of proteins, which exist in a balance of pro-survival and pro-death members. The pro-death members, BAX, BID, and BIML among them, when activated, translocate to the mitochondrial membrane, punch holes in it, and release the signals that commit a cell to self-destruction. Humanin directly interferes with this. Intracellularly, it binds BAX and prevents its translocation from the cytosol to the mitochondria. It binds BIM-EL, it binds truncated BID. In doing so, it holds the pro-apoptotic machinery in check and keeps the cell alive under stress that would otherwise trigger death. This is the mechanism that explains the original discovery. The neurons that survived in that occipital lobe were, in effect, better protected against the apoptotic signals that were destroying their neighbors. The second mechanism is receptor-mediated, and this is what makes humanin a true hormone rather than just an intracellular guardian. Secreted humanin acts on a specific cell surface receptor complex, a trimeric receptor made up of 3 components: the ciliary neurotrophic factor receptor, CNTFR, a protein called WSX1, which is part of the interleukin-27 receptor system, and GP130, a signal transducing subunit shared by a whole family of cytokines. When humanin engages this complex, it activates the JAK2 to STAT3 signaling pathway. STAT3 is a transcription factor with broad pro-survival and anti-inflammatory effects, and its activation is a major part of how humanin protects cells at a distance from where it was produced. There's a second receptor interaction worth noting as well. Humanin binds formyl peptide receptor-like 1, FPR2, and this interaction is thought to be relevant to how it modulates amyloid beta's effects and inflammatory signaling in the brain. The third mechanism connects humanin directly to the insulin and IGF-1 axis, the same conserved longevity pathway I discussed in the clotho episode. Humanin binds insulin-like growth factor binding protein 3, IGFBP3. This is mechanistically important because it places humanin in direct conversation with the growth signaling system that across essentially every model organism studied is inversely related to lifespan. Turn IGF-1 signaling down, and animals live longer. And the correlation runs in the expected direction. In long-lived mutant mice, the Ames dwarf, which has dramatically reduced growth hormone and IGF-1 signaling and lives far longer than normal, circulating humanin is markedly elevated. The peptide sits at the intersection of metabolic signaling and cell survival, which is exactly where you'd want a longevity-associated molecule to be. The fourth area is metabolic, and it's one of the more compelling translational threads. Humanin improves insulin sensitivity and protects the insulin-producing beta cells of the pancreas. In rodent models, administration of humanin, or more often, its potent analog, lowers glucose, improves insulin action, and protects beta cells against the kind of stress that drives them toward failure in type 2 diabetes. Given that metabolic dysfunction is one of the central engines of aging, a mitochondrial peptide that improves insulin sensitivity and is released under metabolic stress fits a coherent picture: the mitochondrion sensing energetic strain and signaling for a protective, insulin-sensitizing response. And there's a fifth thread worth naming: cardioprotection. In models of ischemia-reperfusion injury, the damage that occurs when blood flow returns to a tissue after a heart attack or stroke, Humanin reduces infarct size and protects tissue. This is consistent with its core anti-apoptotic, antioxidative stress activity applied to a setting where a large number of cells are dying acutely and quickly. So to summarize the mechanistic picture, Humanin is a mitochondrially encoded cytoprotective peptide that blocks apoptosis directly by neutralizing pro-death BCL2 proteins, signals through a trimeric CNTFR/WSX1/GP130 receptor to activate protective STAT3 signaling, engages the IGF-1 axis through IGF-BP3, and improves insulin sensitivity. That's a broad and coherent portfolio, and it explains why a peptide discovered in the context of Alzheimer's disease has ended up being studied in diabetes, cardiovascular disease, and aging more broadly. Now let's talk about what the human data actually shows. And here I have to be even more careful than I was with Clotho, because Humanin's human evidence base is thinner, and the gap between the enthusiasm and the clinical reality is wider. The human data falls into two main categories: observational associations and a small amount of genetic and biomarker work. Interventional human data, trials where you give people Humanin and measure what happens, is, for practical purposes, absent. The observational picture is the most developed, and its central finding is consistent with the aging story. Circulating Humanin levels decline with age in humans. Work from Pinchas-Cohen's group at the University of Southern California, one of the leading laboratories in mitochondrial-derived peptide biology, has shown a substantial fall in plasma Humanin across the adult lifespan, with some analyses reporting large reductions between early adulthood and old age. Beyond the age association, lower Humanin has been linked in various cohorts to markers of poorer mitochondrial function and to several age-related disease states. And in the other direction, there's an intriguing signal at the extreme of longevity. The children of centenarians, people genetically enriched for exceptional lifespan, have been reported to carry higher Humanin levels than age-matched controls. It's a small literature, but it points the same way the Clotho data does. Less of the peptide with age, and more of it in those who age exceptionally well. I want to be as clear here as I was with Clotho. These are associations. Observational data cannot establish causation. It's entirely possible that Humanin declines as a consequence of mitochondrial aging rather than as a driver of it, that it's a readout of mitochondrial health, not a lever on it. The centenarian offspring finding is suggestive, but it's a correlation in a genetically unusual population, not proof that raising Humanin extends life. The genetic and biomarker work adds some texture. There's interest in a specific mitochondrial DNA variant within the Humanin coding region that alters the peptide sequence and appears at different frequencies across populations with different disease and longevity profiles, though this remains an area of active and unsettled investigation rather than established fact. And then the interventional data. Here is where I must be completely direct with you, exactly as I was about Clotho. There are no completed Phase 2 or Phase 3 human clinical trials of Humanin administration. There is no approved Humanin product. There is no established human dose, no human efficacy data, and no long-term human safety data for exogenous Humanin as an anti-aging intervention. Essentially, everything we know about what happens when you increase Humanin in a living system comes from cells and from animals, and critically, a great deal of even that animal work was done not with native Humanin at all, but with a synthetic analog, which brings me to a point that matters enormously and is almost always glossed over. Much of the most impressive Humanin data, the neuroprotection, the metabolic effects, the cognitive findings, was generated using a modified version called S14G-Humanin, often abbreviated HNG. It's native Humanin with a single amino acid swapped, a glycine substituted at position 14. That one change makes the analog roughly 1,000-fold more potent than the natural peptide. That is not a subtle tweak. When you read that Humanin protects neurons or Humanin improves insulin sensitivity, there's a very high chance the experiment used HNG at a potency the natural molecule simply does not have. This doesn't invalidate the biology. It's genuinely interesting that such an analog exists and works, but it means you have to read the literature with your eyes open. The gap between a mitochondrial peptide your body makes and a synthetic super analog injected at pharmacological doses in mice is exactly the kind of gap where translation to humans succeeds or fails. And the history here should keep us humble. As I said last episode, longevity biology is littered with compounds that perform beautifully in mice and failed to translate. Resveratrol, telomerase activators, the early optimism around NAD precursors. Humanin at this stage has less human interventional evidence than any of those had at the point they entered clinical testing. The biology is real and the concept is exciting. The clinical evidence is close to a blank page. I want to spend a moment on the neuroprotective and cognitive angle specifically, because it's where Humanin began and where much of the ongoing interest sits. The founding observation that Humanin rescues neurons from amyloid beta toxicity has been replicated many times in cell and animal models. In rodent models of Alzheimer's type pathology, administration of Humanin, and more often HNG, has been reported to preserve memory and reduce markers of neurodegeneration. The proposed mechanisms are consistent with what we've already discussed: blocking apoptosis in stressed neurons, activating STAT3-mediated survival signaling, dampening the inflammatory response to amyloid, and interfering with amyloid beta's toxicity through the FPR2 receptor interaction. There's also work suggesting Humanin supports the survival and function of the cells that maintain the brain's connectivity. The coherence here is real, a molecule discovered because it protects surviving neurons, which turns out to block the specific death pathways those neurons face, is a satisfying scientific story. But I'd apply the same caution I applied to Clotho's cognitive data, only more so. Rescuing neurons in a dish and preserving memory in a genetically engineered mouse, dosed with a 1,000-fold potent analog, are a long way from treating human cognitive aging. We do not have that human evidence. What we have is a mechanistically credible hypothesis with strong preclinical support and no clinical confirmation. Given that Humanin declines with age and lower levels track with worse outcomes, the natural question is the same one we asked of Clotho: can you raise it? And if so, how? Exercise is once again the most robustly supported lever. Physical activity is a mitochondrial stressor in the productive sense. It challenges mitochondria and provokes adaptive signaling, and studies have reported increases in circulating Humanin and related mitochondrial-derived peptides following exercise. The mitochondrial stress logic is clean. You stress the system in a controlled way, and it responds by releasing the protective peptides it evolved to release under stress. Caloric restriction and fasting are the second lever, and they're mechanistically coherent for the same reasons they matter for Clotho. Restricting energy intake activates AMPK, suppresses mTOR, and shifts cells toward a stress-resistant, repair-oriented state, the same metabolic territory Humanin operates in. Animal data supports the idea that caloric restriction raises Humanin, and it fits neatly with the observation that the long-lived growth-signaling suppressed Ames dwarf mouse has high Humanin levels. Human interventional data on fasting protocols, and Humanin specifically, is limited, but the mechanistic rationale is sound. Maintaining metabolic and mitochondrial health more broadly belongs on this list too. Because Humanin is a mitochondrial peptide, the general strategies that support mitochondrial function, regular aerobic activity, avoiding chronic metabolic overload, good sleep, are reasonable ways to support the system that produces it, even if the direct Humanin data is thin. And then the interventional frontier. This is where the synthetic analogs come back in. The existence of HNG and the broader interest in mitochondrial-derived peptides as a drug class means there is genuine pharmaceutical and academic effort going into whether these molecules can be developed into therapeutics for metabolic disease, for neurodegeneration, for cardioprotection. But I'll say it plainly, this is early-stage research, not clinical practice. Anyone offering you an injectable Humanin product today with claims of anti-aging or cognitive benefit is operating far ahead of the evidence, considerably further ahead than even the clotho sellers I criticized last episode. There is no human efficacy data to stand on. So where does this leave Humanin in 2026? Conceptually, Humanin is one of the most interesting molecules in this entire series. It rewrote our understanding of what the mitochondrial genome does. It founded a new class of signaling peptides. Its discovery story, pulled from the surviving neurons of an Alzheimer's brain, is not just poetic, but scientifically instructive because it led directly to a coherent, well-characterized set of protective mechanisms. The anti-apoptotic biology is solid. The receptor signaling is defined. The metabolic effects are reproducible in animals. The age-related decline in humans is documented, and the centenarian offspring association is a tantalizing thread. What we don't have is anything approaching clinical proof. We don't have completed controlled human trials. We don't have an approved product, an established dose, or human safety data for its use as a longevity intervention. And we have the specific complication that much of the compelling data rests on a synthetic analog many times more potent than the natural peptide, which widens, rather than narrows, the translational uncertainty. On the spectrum from credible research target to clinically actionable, Humanin sits earlier than clotho, and clotho itself, as I said, is not yet clinical. What we can say is this: Humanin represents a genuinely novel and scientifically credible idea that our mitochondria speak to the rest of the body through peptides, and that those peptides decline as we age. The basic science is strong, the human evidence is preliminary, and the evidence-based strategies for supporting it, regular aerobic exercise, sensible metabolic and caloric discipline, and maintaining mitochondrial health, are worth pursuing entirely on their own merits, regardless of what the Humanin biomarker is doing. That's Humanin, named as a statement of hope. First discovered in 2001 in the surviving neurons of an Alzheimer's brain, the founding member of the mitochondrial-derived peptides, proof that the small circular genome inside our mitochondria does more than power our cells. It sends messages. A molecule with elegant, well-defined protective biology at the preclinical level and an almost entirely undeveloped clinical evidence base. In the next episode, we'll stay inside the mitochondrion and cover MOTSC, the second major mitochondrial-derived peptide, an exercise mimetic with a fascinating link to metabolic health, and an evidence base that is, in its own way, just as early as Humanin's. We'll do it the same way. What it is, what the proposed mechanisms are, what the evidence actually shows, and what the honest picture looks like. If this series is useful to you, share it with someone who's navigating this space. This field deserves more rigorous public discussion and less noise. I'm Dr. Hausman-Marquis. This is the Science of Ageless Living.